In this video, we will discuss about the CD16 NK-92 expression and its activity validation. So for that, they use uh, Ritu- Rituximab, which is one kind of drug that is used to coat the target cells, mainly the B cells that has the CD20 expression, and thereby it has been detected by the immune cells through the Fc receptors that are present on this Rituximab. So they have used this Rituximab activity or properties to detect CD16 expressions. So how they use this rituximab in this study that we will discuss in this video. So the panel A discussed about the activity validations of this rituximab, whether rituximab is working or not. So how they validated? They checked it on the B cells, that is Raji cells and Daudi cells, which are known to have a high expression of the CD20. And in this we can see that the expression levels are around 98.2% for the Raji cells, 99.1% for the Daudi cells. Then they exogenously expressed CD20 on the K56. In that case also they are able to see the high expressions of this CD20 to around 63.5%. Here this rituximab is conjugated with sci-fi, so they check the signals for the sci-fi. So once validating that this rituximab is working, now they check these CD16 and NK-92 expressions by using this rituximab, as the Fc receptor regions that are present on this rituximab will be detected by the CD16 that are expressed on the NK-92 cells. So compared to the wild type, uh, they can see the expression levels of this. They can see the increased signals for this anti-CD20 sci-fi for the CD16 NK-92, and the first left hand side figure of represents the cDNA CD16 NK-92 expressed cells, and the right hand side of the panel B figure represents the CD16 fused CAR or the CAR CD16 NK-92 expressed cells. So we can see that in the CAR NK-92 expressed cells, the signals intensity is slightly higher compared to the cDNA CD16 transduction NK-92 cells. And these things they have later later verified using the ELISA assay, where they can see that the fluorescence intensity of this NK-92 CD16 cells are high compared to the NK-92. NK-92 CD16s are high compared to the wild type, but NK-92 41BB, which is the CD16 fused with CAR, shows the maximum relative fluorescence intensity. So this indicates that their CD16s are expressed on the NK-92 cells, as the CD16 expressed NK-92 cells will bind to the Fcregion of the rituximab, and the rituximab is conjugated with sci-fi in this experiment, and that is the reason they are able to see high signals compared to the wild type. So these things they later verified with the ELISA also. So these two results giving us the same result, these two different experiments giving us the same results, indicates a point that CD16 and NK-92 express cells are CD16 and NK-92 transduced cells, or the NK-92 transduced cells has high expressions of the CD16, and both in the case of cDNA CD16 and this CD16-CAR transduced NK-92 cells. So, and their activities are also seems to be uh, working, as they can detect the Fc regions of the rituximab, which is essential to trigger the cytotoxicity effect against the B cells. So we will discuss further activities in our subsequent videos. I hope you understood how they validate the CD16 and NK-92 expressions and their activity validations using Rituximab. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.